Hello viewers, welcome to the first video of Biochemistry and Metabolism series. This is a topic picked up from the PG syllabus of Borderland University. So in this video, we will be discussing about classification and structural features of amino acid. In this slide, we will be discussing about characteristics of amino acid. So we all know that proteins are involved in various functions like fighting infections and cell division. Now what are these proteins made of? These proteins are made of amino acid. Now what is amino acid? Amino acids are compounds consisting of carbon, hydrogen, oxygen and nitrogen. It serves as monomer and a building block of proteins. As you can see in the structure here, general structure of amino acid contains alpha carboxyl group, alpha amino group, hydrogen and an R side chain. So here alpha carboxyl group act as an acidic and can donate proton, while alpha amino group act as an basic and can accept proton. This point says there are 22 standard amino acids which participate in protein synthesis. Various amino acids differ with respect to R group attached to the alpha carbon. So all these standard amino acids are different from each other based on what R group has attached to the alpha carbon. As you can see in the structure of glycine and in the structure of proline. In the structure of glycine, each atom has attached to alpha carbon as R side chain, while different R group has attached to alpha carbon as side chain in proline. So based on what R group is attached to alpha carbon, all amino acids are different from each other. Next point, glycine is the simplest amino acid with hydrogen atom as side chain. Glycine is considered to be the simplest amino acid among those 22 standard amino acids because in glycine you will see that each atom has attached to alpha carbon as this side chain, and you will see in different amino acid, in other amino acid there is no R group which has each atom as their side chain. So glycine is considered to be the simplest amino acid. Next, proline is secondary amino acid also known as amino acid. Here proline is considered to be the secondary amino acid and also known as amino acid because in proline you will see R group is attached to alpha carbon as well as to alpha amino group. So for this, proline is an unique amino acid which is also known as secondary amino acid and also amino acid. Here this point says at physiological pH 7.4 amino acids are present in zwitterionic form, which means it has both negative and positive charge. Zwitterion means a molecule consisting of both these charges. You will see that all amino acid will have a specific pH where it will be existing in zwitterionic form. You see here in this structure, when pH is equal to its intermediate pH, this carboxyl group is acting as an acidic. As I have said in the previous slide, carboxyl group acts as an acid and can donate proton. So here this carboxyl group has donated its proton and have become negatively charged, while this amino group can act as basic and can accept proton and it has gained positive charge. So here both charges are present, negative as well as positive. So this amino acid is existing in zwitterionic form and the net charge of this amino acid will be 0. Next point says at very low pH the overall charge of amino acid is positive. So when pH is very low than its intermediate pH, what happens is in this case this carboxyl group will be protonated. It will not donate its proton and it will be uncharged, while this amino group will accept proton and it will become positively charged. So overall charge of this amino acid will be positive. Next point, at high pH the overall charge is negative. So here you will see that when pH is higher than its intermediate pH, this carboxyl group has become deprotonated and has donated its proton and become negatively charged, while this amino group also become deprotonated and it remain uncharged. So overall charge of this amino acid is negative. Next point says amino acid are amphiprotic as well as amphoteric. Amphiprotic means it can accept and donate proton, and amphoteric means it can either act as acid or base. So amino acid can act either acid or base, and it can also accept and donate proton. Here, this point says all amino acids except glycine are optically active and is chiral molecule. Now, what does it mean by chiral molecule? You will see in the structure of glycine and in the structure of alanine. In structure of alanine, alpha carbon has attached to 4 different substituents, while in the structure of glycine, alpha carbon does not have 4 different substituents. So, alpha carbon which have 4 different substituents is known as chiral center, and this amino acid is known as chiral amino acid, while alpha carbon which does not have 4 different substituents is not known as chiral center, and this amino acid is known as achiral molecule. So all amino acid except glycine will be optically active and is chiral molecule. Now we know that all amino acid except glycine are chiral amino acid, and this chiral amino acid can exist in two configurations, and these two configurations are called enantiomers. In DL system, all amino acid except glycine which are ribosomically incorporated into proteins exhibit L configuration. Now, difference between L configuration and D configuration is that in L configuration, alpha amino group is attached to alpha carbon from the left side, while in D configuration, alpha amino group will be attached to alpha carbon from the right side. Now in RS system, you will see that all L amino acid from proteins will have S absolute configuration except for L cysteine, which will have R configuration. So here it is the classification of amino acids. Amino acids are classified based on the side chain. Amino acid with non-polar side chain are hydrophobic. By the word itself says water hating. Hydro means water and phobic means hating. So these are water hating amino acids. These are glycine, alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine, and tryptophan. Whereas amino acids with uncharged polar side chain are hydrophilic. By the word itself, it means a water-loving. Hydro means water and philic means loving. So it is a water-loving amino acid. These are serine, threonine, cysteine, asparagine, glutamine, and tyrosine. So next is amino acid with charged polar side chain at pH 7. This can be positively charged R group and negatively charged R groups. Positively charged R groups are lysine, arginine, histidine, and negatively charged R groups are aspartate and glutamate. So in this slide, it is about essential and non-essential amino acids. Essential amino acids are those amino acids that cannot be synthesized in sufficient amount by our body, and for the proper functioning of our body, we require or we obtain it from other dietary external sources. So these amino acids are arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Whereas non-essential amino acids are those amino acids that can be synthesized from amphibolic intermediates by itself in our body and are not required for any other dietary sources. So these are known as non-essential amino acids. They are alanine, asparagine, cysteine, aspartic acid, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. So that's all for this video, and if this video was helpful for you all, please like it and subscribe it. And in upcoming video I will be discussing about peptide and peptide bonds.